Obrigado. Primeiro eu quero dizer obrigado pelo convite. É prazer para mim estar aqui. Eu adoro o Brasil e este curso é muito bom. Now, sim? Não precisa? Obrigado. Então, eu tenho uma tarefa difícil. Por quê? Ontem à noite, todos foram bebendo e bailando, sim. E então eu tenho que acordar todos. Eu sei que inglês é uma língua difícil, mas desculpe, meu português é muito ruim e o meu sotaque é pior do que Mario Stevenson. Sim, é verdade. Então eu vou falar em inglês. Okay. Sorry about that. So we're going to be talking about the adaptive defenses to infection with virus. And this lecture I'm going to give you, instead of just focusing on some of the latest and greatest stuff from my lab, I thought it would be more useful, especially to the students, to really give you a broad overview of the basic mechanisms of management presentation. And these are really going to deal with how we recognize viral infection. So when a virus enters the body, there are, as you know, multiple lines of defense that the body can use to deal with this. Some of them, like antibodies, are effective when the virus is still in the extracellular space. They can neutralize it. But once a virus enters a cell, infects it, it's taking over its biosynthetic machinery, these kinds of mechanisms are no longer effective. And in this situation, the major adaptive immune response that helps us deal with infected cells is the CD8 T cell. So the job of this cell is to recognize that this cell is infected. It's a viral factory. That's a bad thing for the body. And upon that recognition, one of the things that it will do is to kill the cell, which will eliminate the viral factory, which is good to try to restore us back to sterilizing immunity. So this is true for essentially the majority of viral infections, and it's particularly true also, we know, for HIV/AIDS. So this is obviously a viral disease. We know that CD8 immunity is important for many reasons. So one thing, if we look at the natural history of a patient and follow their viral loads, there's a correlation with the control of viremia with the appearance and duration of CD8 T cell responses. There's also very strong genetic evidence that I'm going to show you. Actually, I'm not going to show you this. There's strong genetic evidence that the virus evolves. And that evolution, you can see the imprint or the effect of CD8 T cell recognition on the virus driving the virus evolution, its mutation away from recognition. And this is, of course, also important in the subject because AIDS patients get infected with a lot of other viruses. And again, this is the major pathway that's going to recognize them. So coming back to this point, this is a little video here. This is a virus-infected cell. This is a CD8 T cell that has recognized this as being virally infected. And what it does is it makes a tight adhesion, and then it's going to deliver a lethal hit to this cell that's going to cause it to die. And so biologically, what the system is trying to do is to eliminate these infected cells because they're a source of the pathogen, right? So how is it that the CD8 T cell is actually able to recognize that this is not a normal cell, but one that's virally infected? And what it's actually looking for here is this structure shown here. So this is one of the peptide binding receptors that's encoded in the major histocompatibility complex. These structures decorate the surface of essentially all cells in the body, and T cells circulate looking to see if they can find one of these. Okay. And this is actually a T cell's view of the molecule. So the molecule is sticking up out of the membrane, and we're looking down on the surface of it. And what the T cell is actually looking for is this little structure shown here, which is an antigenic peptide. Okay. Now, the basic pathway that I'm going to walk you through is shown in this little cartoon here. So all cells will transcribe a subset of their genes, and those will get translated into proteins. All proteins in cells are eventually turned over. Different rates, but they're turned over. And as an intermediate, as these are getting broken down, one generates peptides. Now, a fraction of these peptides, this is a process occurring in the cytosol and nucleus, a fraction of these peptides get transported through a transporter I'll tell you about into the lumen of the endoplasmic reticulum, where these peptide binding receptors are assembling. And so they assemble here, and they'll bind the peptides that they have specificity for, and then these molecules will migrate, be exported to the cell surface, so that they display on their surface a sampling of the peptides that come from the proteins from your expressed genes. Now, hopefully in all of us in this room at the moment, in healthy situations, all of the proteins that are getting degraded and the peptides that are getting made come from our normal proteins, okay? They should be there. And so while the cells display that information, T cells can come and look and see that these peptides are really normal ones, and they shouldn't do anything about that. And the reason that they know that is, I'm not going to go in today, but is the basic mechanisms of thymic selection and T cell self-tolerance. Okay, so this is normal physiology. Now, if a cell was actually synthesizing something that's foreign, and if you think about it, the number of times in nature that your own cells are going to synthesize something that's foreign is very limited, right? You're either transcribing somebody else's genes, like a virus, you've mutated some of your genes on the way to becoming a tumor, also a dangerous situation for the body. And in nature, really, those are the only two circumstances. Clinically, we also cause that in patients when we transplant tissues, but I'm not going to get into that today. So if we have a situation where a virus enters a cell, some of the viral genes are going to get transcribed and translated, and some of that will get made into peptides, and some of that information will come out on the cell surface. Some of the viral peptides bind to the class I molecule. And this now allows the CD8 immune system to see that this cell is expressing something that it shouldn't, it's dangerous, and it should be eliminated. Okay, claro? Okay. So I want to start talking in a little bit more detail about what these peptide binding receptors are. And so this is a couple images of them. The first one is a very simple sort of stick figure cartoon. And to orient you, this is the cell membrane in dark gray. The interior of the cell is down here, and this is the extracellular space. And so what you can see is this is a two-chained molecule. The larger chain, or the heavy chain, is called alpha. It's got multiple domains. And it has a second chain that's not transmembrane, that's smaller, so we call that the light chain. And the other name for that is beta-2 microglobulin. Okay, so it's two-chain structure. Now on this side, we've kept the color coding. This is what the actual structure looks like from X-ray crystallography. This is the alpha carbon backbone, really starting from here on up. And so you can see the membrane distal domains here form what at the time this was discovered was really a unique structure. We see that nature has used it in some other places as well. But what you can see here is that there are two alpha helices projecting up, right? And they're sitting on a beta-pleated sheet. So what I always tell our medical students when they try to remember the structures, they should think about this as a hot dog bun, right? A cachorro quente, see? Sitting on a barbecue grill, okay? And what do you think the hot dog is? It's the peptide, right? Okay. Now, this heavy chain, this alpha chain here, I want to tell you a little bit about the genes that encode it, because it turns out that this is important. So these genes are encoded in a locus on chromosome 6 in humans, chromosome 17 in mice, called the MHC, which stands for Major Histocompatibility Complex. And that name comes because their discovery in part was made based on transplantation, histocompatibility between individuals. And this was a major determinant. Okay. So I want to focus on the human MHC, which we call HLA, shown down here. This is the mouse up here, for those of you that are interested. And the genes that we're going to talk about are the ones here that are colored in pink. So these are known as HLA because of the MHC locus. So they're called HLA A, B, and C. Okay, simple. 3 genes. Now, these genes, I just want to remind you, are encoding this yellow or the heavy chain. The second chain, beta-2 microglobulin, is encoded elsewhere. It's not in the MHC. It's a single gene. And in the species, the beta-2 microglobulin is not really polymorphic. So that means that all of us basically have the same gene sequence. As I will tell you, that turns out not to be true for the heavy chain. Okay. So, I've said there are how many class 1 genes? Por favor, ¿cuántos? 3, right? And it turns these are polymorphic, and so you can get 3 from your mother and 3 from your father that can be different. So the maximum number that you could have is how many? 6. Very simple, okay. How many potential antigens are there? They're huge numbers, right? So how can only 6 class 1 molecules present all antigens? And this is something that really confused the field for a number of years. And the answer comes from actually examining how the MHC molecules are binding the peptides. So I've shown you this image before. This is the same image taking away some of the space, just showing the alpha carbon backbone. This is the peptide that's bound. So part of the answer is that these molecules don't bind the whole protein. What they bind is a small piece of it. So how does that allow these 6 different molecules to bind all peptides? So to consider that, I want to look at what a peptide structure is. This is just a little cartoon of a dipeptide, 2 amino acids linked together through an amide bond. This would not be long enough to bind to a class 1 molecule, but it will illustrate the point I want to make. And so these are 2 amino acids that are linked. And if you look, each amino acid, most of what's present there is the same. The only thing that's a different amino acid to amino acid is the side chain, right? So if this is a methyl, it's an alanine, a hydrogen, a glycine, et cetera, et cetera. So I think you could imagine that if you were to design a receptor that only bound the common parts of amino acids, then you could bind everything, all peptides, right? And to a first approximation, that's something that the immune system tried. So if you actually look how a peptide binds to an MHC molecule, there are a lot of significant molecular contacts with common features. So they bind the free N-terminus, the free carboxy terminus, they make hydrogen bonds with the main chain atoms, et cetera. And this gives a lot of the affinity of binding. Now, it turns out that the immune system couldn't quite get enough affinity from just holding on to the common regions. It had to bind a couple of side chains. And so I'm going to illustrate that here. This is a typical size of an MHC class 1 binding peptide. I'll come back to that. One is 9 residues. And in the hot dog bun, so in this peptide binding groove, there are pockets in this particular MHC molecule that will bind the side chain in position 2 and the side chain in position 9. And these pockets will bind only one or maybe a couple of related amino acid side chains, but not the others. And so that constrains what can fit this molecule. But all of the other 7 residues here, the side chains aren't bound, and so those can vary between any of the 20 amino acids. So if you do the math, if we constrain 2 positions but allow the others to vary between any of the 20, this is a factorial, you'd be able to bind a single molecule can bind a very large number of peptides, but not all. Okay, Antígenos? Okay. So, and it turns out that each of the different class 1 molecules, the HLA A, B, and C, bind different side chains. And so the HLA-A molecule can bind a very large number of peptides, not all. HLA-B can also bind a very large number of peptides, different than the A, but still not all. And so with this mechanism, with the 6 different MHC class 1 molecules, it's possible for the immune system to cover a major part of the antigenic universe in cells. Not all, and that can lead to problems, but still much of it. Okay. So I did mention to you that the MHC class 1 heavy chain is a polymorphic gene, and it turns out that it is the most polymorphic set of genes that we have in the human species by far. So at this point, the field has identified over 10,000 different alleles for class 1 molecules. And this is still an underestimate. So what that means is that my DNA coding sequence for HLA-A is different than yours, most likely, which is different than your neighbor's, et cetera, et cetera. So there's huge variability in the species. Now, I want to point out that it turns out that this is true, in certain populations, certain alleles are more frequent. That's sort of true of all genetics. And so if you look in the United States, in the Caucasian population, particular forms of HLA, like HLA-A2, tends to be more frequent than you would find, let's say, in Asia, et cetera. Okay. So a lot of variability in the DNA coding sequence, and also, therefore, in the amino acid sequence. So where are the variable residues, these polymorphic residues? And we're going to look back at the structure. So this shows the molecule standing up. This is looking at the T cells view. We've moved the molecule down, so we're looking on the surface of it. And every one of these red dots is a position which is polymorphic in the species. Okay, and you can see them clustered here along the alpha helix and down here on the barbecue grill. So what are these polymorphic residues going to affect? So if we change the molecular shape by changing the amino acids in these areas, what is this going to affect? What binds there? Peptide, okay? And so what this means is that the various alleles are going to have different specificity for different peptides, right? Make sense? And what else is going to bind here? T cell receptor, okay? And if you look here, up on these alpha helices that are on the top of the molecule, there are lots of variable residues. And so it's going to turn out, as you probably know, that T cells are going to care about this. And this results in the phenomenon of MHC restriction. A CD8 T cell will be specific not only for a peptide, but the particular MHC allele as well. Okay. So influence T cell receptors and peptides. So why do you care about this? Why am I spending time telling you about this? And the reason is that this has important implications clinically and in biology. So one of the implications of this is that different individuals that have different MHC molecules, different forms, different alleles, are going to be presenting different peptides, and therefore they may respond to different antigens. And this is clearly true. Okay. And this will have implications if you're trying to develop vaccines, particularly folks that think of peptide-based vaccines. And this can also influence, if it's going to influence what the immune system is able to recognize and respond to, it may influence the course of disease, right? Should make sense. And I'm going to show you, this turns out to be true, and there are many examples of this. One I'm going to tell you about here, many of you may know about this, is in the area of HIV. So about 7 years ago, this study was done, and this was a study of the elite controllers of HIV. So I think most of you know that most people that get infected with HIV untreated will progress and develop AIDS and ultimately die. But there's a small subset of individuals who get infected. We can clearly have evidence that they're infected, but they control the HIV. They never progress to AIDS. And so there was a lot of interest in trying to understand what was it about this small subset of individuals that allowed them to actually control the viral infection. Because if we knew that, there might be some interesting clues as to what we could do. And so there was a very large genetic study done looking across the entire genome at single nucleotide polymorphisms in about almost 4,000 patients. And when they analyzed this data, there was only a single gene, or said maybe more correctly, a single set of related genes that were found. And so this shows all of the chromosomes. And here is the one hit, which genetically mapped the elite controllers. Okay, and what these turned out to be were various forms of MHC1 alleles. Okay, and so the conclusion here was that one of the principal genetic determinants of controlling HIV were the polymorphisms of MHC class 1. Okay, so this stuff is important. And I've also not told you another reason this is important is that the polymorphisms in MHC, this is true for MHC1 and particularly true for MHC2, are also the strongest genetic determinants in almost all autoimmune diseases. Autoimmune diseases have multiple contributions from multiple genes, but this is the strongest. Okay. So I just want to summarize what I've told you. The MHC class 1 molecules have a groove that allows them to bind peptides. The peptides are bound via some of their common characteristics, the N-terminus, the C-terminus, the alpha carbon backbone, and a couple of the side chains. And this permits a single molecule to bind a large number of peptides, not all, and the polymorphisms in this molecule affect the binding surfaces and impart specificity. And all of this is important to the subject of this course here, which is HIV. Okay. Okay, now I want to turn and tell you a little bit about how the class 1 molecules actually form. And so these are membrane molecules. So we're looking here in blue, this is the endoplasmic reticulum, and out here is the cytosol. So these are synthesized on rough ER, and the class 1 heavy chain is co-translationally transported into the lumen of the endoplasmic reticulum, as is the light chain, and that folds and associates with a number of chaperones that sort of keep it nicely folded and ready for a peptide. And this molecule tapesin that I think I'll tell you a little bit more about, and this TAP transporter I'm going to tell you a little bit more about. And this is the location where it's waiting for peptide to go to the cell surface. So I want to tell you now a little bit. So these class 1 molecules are bound in the endoplasmic reticulum. Initially, they have no peptide, and they're needing to get it. And so I'm going to talk to you a little bit here about how peptides actually get to the class 1 molecule. And the issue is here that the MHC class 1 molecule, as I've just shown you, is formed in the inside of the endoplasmic reticulum. And the peptides that are generated in the cytosol nucleus. Remember I told you that in one of the first slides, that's where the degradation occurs. And so the immune system has a problem because the peptides are in the wrong place And between them and the peptide binding receptor is a highly hydrophobic membrane. And energetically, there's no way that a 9-residue peptide can actually get across this by diffusion. And so to deal with this, the immune system evolved a peptide transporter called TAP. It has 2 chains coded by 2 genes. These are found in the major histocompatibility complex. And so they're expressed, they form a channel here that pumps in an energy-dependent way the peptides from the cytosol into the endoplasmic reticulum. Okay. So you need this for the pathway to work, and you only have one of them. So it turns out that it's a fairly non-specific transporter. It can transport a majority of peptides that are the right size, 8 to 17 residues. If they get longer, it doesn't work so well. And it has a little bit of specificity. So some peptides it doesn't like to transport. And a consequence of that is it's going to limit some of the peptides that would be available for display. It's an unavoidable consequence of the system. So it's a little bit of a filter. Okay. So we've talked about the peptide binding receptor. We've talked about this peptide transporter. I now want to turn to how the peptides are actually generated. So when the immune system evolved this system, which was fairly late in evolution, so this really came up at the same time as adaptive immunity, which is late in early vertebrate development. The immune system needed a source of peptides from a cell's expressed genes, and it looked around and it found that cells were already making peptides. It didn't need to reinvent this process. So as cellular proteins are normally degraded, as I've told you, one of the intermediates are peptides, and so this is part of the normal catabolic process. So the proteins that are expressed in your cytosol and nucleus are degraded by a system called the ubiquitin proteasome system, which generates peptides, and these peptides can then be further degraded by other peptidases to amino acids, which are useful for protein synthesis or energy. So the actual protease that cleaves these is shown here. It's a structure called the proteasome. This is a large particulate proteolytic particle. It's found in the cytosol and nucleus of all nucleated cells. This has actually been opened up. It's been cut in half so you can see the inside of it. And you can see there's an aqueous channel that runs through this. And in the center of this channel, these little gold things are the active sites. And so it will take a protein, thread it here, it hits the active sites. These are cleaved, and peptides then exit. Okay. So I want to tell you some of the evidence that this is actually a key pathway. So this is some data we published 20 some odd years ago, but it illustrates the point. So what I'm showing you here is an assay for the appearance of a peptide MHC complex. We're actually using a T cell that will get activated if it sees this complex. And what we're doing is taking a cell and we're going to express in it a foreign antigen. This happens to be chicken ovalbumin. We're using a vaccinia virus to express this. And so if we express this, the cell starts to synthesize ovalbumin, peptides will get generated, these will go into the class 1 molecule, come out on the cell surface that we can read out. Now, years ago, we and our collaborators developed very specific inhibitors of the proteasome, and one found if you inhibited the proteasome in cells, you could completely block this presentation. Okay, very clear result. Now, I could tell you could get the same result if you poisoned the cells, because you need metabolic activities. So is this really just blocking the generation of this? And so one of the ways that this was looked at is in the case of the ovalbumin, we know the exact peptide sequence the cell needs to display on the surface, needs to generate and display on the surface. It's a sequence here called synfickle. So what we've done in this side of the experiment is instead of asking the cell to make the synfickle from the whole ovalbumin, we've given it ready-made, and we've done that by making a synthetic gene that encodes synfickle and express that in the virus so the cell just actually synthesizes the peptide, right? Make sense? Okay. And so when we do that, the peptide gets presented, and if we treat with a proteasome inhibitor, there's no inhibition. And so what that tells you is that you need proteasomes if you're going to present a peptide from a whole protein, but after the system has cleaved it, you don't need the proteasome anymore, and that implicates this molecule, this protease, in generating these peptides. These inhibitors don't affect anything downstream. And so this sort of data and much more that's in the literature has led to the conclusion that proteasomes are necessary for the generation of a majority of peptides for MHC class 1 molecules. Now, so the immune system clearly used this phylogenetically older catabolic system, but also modified it. And I'm going to tell you about that in the next slide here. So this is my little cartoon of the proteasome. I've told you that there are active sites in the center of it, and I'm representing that here by scissors. There's 3 of them, beta 1, 2, and 5. It turns out that there is a duplicated set of these genes, a slightly different sequence, that we call beta 1I, 2I, and 5I. And these alternate versions of these scissors, 2 of which are encoded in the MHC, by the way, are expressed constitutively in dendritic cells and some lymphoid cells. And in all other cells, this can be turned on in the presence of inflammation, particularly by interferons. Now, when these express, what they do is they outcompete the normal scissors from incorporating, so you form new proteolytic particles that have new active sites. And these are called immunoproteasomes. And we know from studying these that when a protein goes through this one versus this one, you get different sets of peptides. Some are the same, but many are different. Okay. So how important are these? A number of years ago, we generated a mouse which knocked out all of these new subunits. This was in the pre-CRISPR era, so this was actually technically challenging, but we did it. And then we asked, well, how different are the peptides in this mouse versus one that has immunoproteasomes? And the answer is that they're very different. So this was done in the mouse, so these are 2 of the mouse class 1 molecules. And we found when we characterized several hundred of the peptides from mice that lacked immunoproteasome, that's in pink, or from ones that just had immunoproteasome, that about 50% were qualitatively different, and many of them were quantitatively different. So this is a huge difference. And you could imagine that that's going to have biological consequences, and it does, because if you take an animal that lacks immunoproteasomes and infect it with a virus, in this case LCMV, and analyze the CD8 T cell response to that viral infection, it's markedly changed relative to a wild-type animal. 9 of the 11 epitopes, or the presented peptides, are presented at different levels. Most of those are markedly reduced. There's one that's elevated. Okay. So the point of what I've just been telling you here is that in the surveillance pathway, the first thing that's going to happen is that the cell expresses genes. Those are degraded by proteasomes or immunoproteasomes, and that gives us a library of peptides. Okay. Now, is that the end of the story in terms of generating these? And the answer is no. And I want to come back and look at the structure of the MHC class 1 molecule. So here you can see the peptide that's bound. And it turns out that the class 1 molecules, the peptides that can bind to any class 1 molecule are remarkably uniform in length. And the reason for that is that this hot dog bun, this groove is closed, okay? So a long peptide can't fit in it. It's going to flop out. And not only is it closed, but there's a pocket that binds the free N terminus and a pocket that binds the free C terminus on the peptide. And so a long peptide can't fit in those pockets. And so this contributes a lot to the binding affinity. And so for most human class 1 molecules, the peptides that are bound are either 9 or 10 residues in length, and for mouse, it's either 8 or 9. Okay. So does the proteasome actually make peptides of the right size? And one of the ways that you can look at that is to take purified proteasomes. You can feed them your favorite protein, allow them to degrade, collect the peptides, and look at their size. And this is what you would find. So what we're looking here is the size of the peptides on the x-axis and their abundance on the y-axis. And these are 3 different proteins. And what one can see is you get a log normal distribution. And I put this little red box here to indicate ones that are the proper length to bind to a class 1 molecule. So you can see the proteasome makes a few. It makes a bunch that are too short. Those are lost to the system. And it makes a larger number that are too long. So these can't stably bind, but they could be precursors, right? So are these long peptides actually precursors? And one can look at that by injecting or expressing these long peptides in cells. And when you do that, you find that the relevant peptide is actually generated and presented. And if you characterize the systems that do this, it turns out that the proteasome is the only protease in cells that can make the proper C-terminal cleavage. But peptides that are long on their N-terminus can still be used. These residues can be taken off. And that's done not by the proteasome, but by other peptidases. So the key points here is in order to generate peptides from the expressed genes, the immune system utilizes this normal housekeeping cellular machinery to degrade the protein. The cleavages, the initial cleavages, are made by the proteasome, resulting generally in long precursors. In this process, the proteasome makes the proper C-terminal cleavage, but other peptidases can cleave the N-terminus of the peptides. And the majority of these peptides are generated in the cytoplasm, and as I've told you, the TAP transporter will transport them to the class 1 molecule. Okay. So what do we know about these other peptidases that we're going to need to convert these precursors into the presented peptide? And so it turns out that much of this trimming, this removal of the N-terminal amino acids, is occurring in the endoplasmic reticulum. And the critical enzyme that does this is an aminopeptidase, so peptidase that takes off from the amino side of a peptide. And when we discovered this, we called it ERAP1, and when Nilab Shastri discovered this, he called it ERAP, and you'll see both of these in the literature. And I should point out that the mouse only has one of these, but the human has an ERAP1 and also an ERAP2. So how important is this? So a number of us knocked out ERAP1 from the mouse genome, and it does make a difference. So what I'm going to show you in this slide here is if we've infected a mouse with the virus LCMV, and LCMV is highly immunogenic, you get a very robust CD8 T cell response, and those CD8 T cells are responding against multiple viral antigens or peptides. And this shows 6 of the peptides that are recognized. This is in a normal mouse, and wild-type mouse. And what one sees is that some peptides are recognized more strongly than others. This is an immunodominance hierarchy. It's very reproducible. It is very hard to change. Most knockout animals, if they have a response, it's the same. But if you take a look at an ERAP1 knockout, what you can see is that the hierarchy is changed. Most of the epitope responses are down. One is increased. So clearly, ERAP1 is contributing to responses. It's the dominant ER aminopeptidase that does this, and it's affecting specificity. And if it's affecting specificity, you might expect that that could have some biological consequences, and that's true. So it turns out that there are polymorphisms in ERAP1 in the human species, and this is becoming increasingly associated with linkage to diseases, particularly autoimmune diseases and some other responses. And so, for example, in the disease ankylosing spondylitis, which is an autoimmune disease where the body attacks the spine and causes a debilitating arthritis, the strongest genetic component, where do you think that's encoded? Por favor. MHC, yes, HLA-B27. The second strongest genetic polymorphism that's involved is one in ERAP. Okay. So this is basically what I've just told you. This is an important peptidase in vivo. It has specificity to influencers' responses. Okay. Now, the other thing I want to tell you about ERAP1 is that molecularly, it turns out to be a fascinating enzyme. It was really unique as an aminopeptidase when we and others discovered it. And one of the things that's very unique about it is that it trims peptides with a molecular ruler. And so what I mean by that is if we take a peptide that's 11 residues in length, and that's too long, right, to bind to a class 1 molecule. So ERAP1 will begin cleaving residues off of the N-terminus to shorten it down. And what it does is it will do that robustly until the peptide reaches a length of 8 or 9 residues in length. Now that number, 8 or 9 residues, should ring a bell for you, because that's the exact size that the MHC class 1 molecule likes to bind. So how does it do this? And in the last several years, the field has obtained molecular insights into how this works. So as all proteases, ERAP1 has an active site. And in a laboratory, we can measure how active that is by putting in a substrate. And the convenient one that we and others use is just a simple dipeptide mimic. So we can take an amino acid like leucine and attach to it a fluorophore, AMC. And when this is bound in an amide sort of bond, this is not fluorescent. But if you cleave the amide bond, it generates fluorescence. So this is a very convenient way to measure the activity of the enzyme. And one of the things that was found was that if you put the substrate in with ERAP1, that's not very active, but if you add a peptide at the same time, you get activation of the cleavage. And it turned out that the peptides that could do that were ones that were small. They had to be 7 residues or less. And so what this implied was that when peptides bound, there was an allosteric activation, and the activation site was probably about 8 residues away from the active site, because peptides that were 8 residues or longer began to compete for this leucine AMC, indicating that this was the likely molecular distance. So Larry Stern in my department and one of his graduate students crystallized ERAP1. This shows the structure here. And this shows a molecular dynamics modeling. And this molecule has a lot of flexibility. As you can see, it can fold on itself. The active site is found down here. And this sort of model that of this flexibility, this is actually done in a computer, but it turns out that this is probably correct because the molecule has now been crystallized in both the open form and in the closed form. Now, what's very interesting about this is if you look at what happens during this molecular movement down at the active site that's going to be down here, this is what you see. So we're looking at very high power. This is the active site down here. This is the zinc molecule. And this is the open conformation, and this is the closed conformation. So in the open conformation, what you can see here is there's a tyrosine side chain that's pointed away from the active site, okay? Now this tyrosine is important to coordinate a water during the proteolytic attack on the peptide bond here. So in the open form, it can't do that, and this is a relatively inactive enzyme. But what happens when the structure closes, this tyrosine rotates and now points into the active site, which will make this an active enzyme. And so the idea here is that if you have a long precursor here, it's long enough to hit this allosteric activation site and stick its other end in the blades and get trimmed. And so it can hit on, get cleaved, hit on, get cleaved. And that works until it gets too short that it can't hit the allosteric activation site. So once it gets to 8 or 9 residues, the enzyme will slow and stop. And so this is a very elegant mechanism to really make peptides of the exact size that you want. Okay. So, now, large proteins typically contain a number of sequences that potentially can bind to a class I molecule. But in nature, if you actually look, only a few particular regions of a protein tend to be presented well. And this is almost certainly a consequence of whether these peptides are actually cut out properly. So are they made or are they destroyed? And so this processing will really influence the specificity of responses. And unfortunately, we know some about what the specificity is, but we don't know of all of the various components. And as a consequence of this, our ability to really predict accurately epitopes is not perfect. Okay. Now, another thing I want to tell you about is that the immune system has been, viruses have been co-evolving with the immune system. It's not in the virus's self-interest to actually allow this system to detect it, because if that happens, then the immune system is able to eliminate those cells that are producing the virus. And as a consequence of this, viruses that can, a subset of them, have evolved mechanisms to try to evade this detection. And I've illustrated some of the examples here on this cartoon. These examples here are all large DNA viruses, herpes family. And so we're going to look at this here. So this is the basic pathway I've talked to you about. A cell expresses its genes, transcribes and translates these into protein. The proteasomes, immunoproteasomes will generate peptides. The TAP transporter will take these into the lumen of the endoplasmic reticulum. This is where your class I molecules are getting synthesized. They bind the peptide, they go to the surface. So, CMV, cytomegalic virus, has evolved a protein that when the class I molecule begins to form in the endoplasmic reticulum, it takes that class I molecule and pushes it back into the cytoplasm, where it gets degraded. So while you're making viral proteins, you don't have the peptide binding receptors to display them. Harder to see the virus. CMV and herpes simplex virus have encoded proteins that block the TAP transporter, okay? So you're making the class I, you're making the peptides, but they can't get together, et cetera, et cetera. And so you might notice that all of these viruses that are on the slide here are ones that if you get infected, and most of us are, these are friends for life. We don't get rid of them. And we think this is one of the reasons, not the only reason, but one of the reasons for this. Now, it's common with these viruses because they've got a lot, they have a lot of DNA, so they can afford to encode some of these things, but this is also true in simpler viruses. And specifically in terms of this conference for HIV, because it turns out that the NEF protein, which is a multifunctional protein for HIV, one of the things that it does is that when the class I molecules bind a peptide, and they're going to go to the cell surface to display it, NEF is capable of hijacking those class I molecules. So they bound their peptide, they're heading to the cell surface, but they get hijacked where they go to lysosomes, which are catabolic vesicles in cells, and get degraded. Okay, so I think I am going to end here. And what I've tried to accomplish, and I hope this has been clear, is to really trace this pathway by which cells, the various genes that are expressed in cells, can actually be monitored by the CD8 T cell immune system, with the goal of attempting to eliminate any cell that's synthesizing something that's foreign, what the various machinery is, what we know about it, how that influences specificity, and the course of health or disease in people with various genetic variations in this. So I will stop here and be happy to answer any questions.